there are a few announcements now. There's like just one announcement now. We will be conducting the quiz competition now in the tea break. So all those who are participating in the quiz are required to stay back. The rest of you can break for tea, and then we will meet back in 30 minutes. Okay, so you are not supposed to tick anything on the questions. You have an answer key given at the beginning, and you only need to write the answer key there. Huh. So yeah, so okay, so whatever you have ticked, you have required to write the letter on the answer box, because we will not be considering anything that you have ticked. Okay, so you have the answer space for the answer key at the top of the page. So for the final session of the day, we have with us a moderator, Dr. Amit Awasthi. He is a senior professor at the Translational Health Science and Technology Institute in Faridabad. His research interests primarily lie in the areas like immunology, infectious diseases, and molecular biology. Our speaker for this session is Dr. Samir Sangitrao. He is the Vice President and Head of Regulatory Affairs and R&D Quality Assurance, Biologics, Zydus Research Center, Ahmedabad.
This is the last session, and this is my privilege to introduce Dr. Samir Sangitrao, Vice President and Head of the Regulatory Affairs and R&D Quality Assurance, Biologics Zydus Research Center, Ahmedabad. And he will be talking to us today about good lab practices. So now the stage is of yours, sir, please. Thank you.
So, good evening, everyone. First of all, I would like to thank Professor Dalai for inviting me for this lecture. And today I will be talking on quality in R&D. So, guys, this is the last lecture. But if you are joining industry, one of the most important lecture. Because first thing that we teach everybody who joins industry is quality. Not that quality is only important for industry, but it is also very crucial when you do academics, when you do research, when you do MSc, PhD, because that's where you are getting trained, and it is very apt for discussing quality at Nirma Institute. So let's begin. So I'll be touching base on the quality, discovery and R&D, criticality of quality in R&D, guidance for good practices in R&D, key resources and how quality aspects are taken care in that, role of quality assurance department, data integrity, which is the buzzword in industry right now, and I think it will be also a buzzword in the publication sector, because when you're doing academic research or university, it is important that you have to take care of plagiarism and data integrity, that you really have the experimental data behind you. Also, from the point of view of patent, data is very, very important. So if you have proper documentation, proper data written, and you can prove that, then you get the patent for yourself. So the talk is going to talk about all of this. People who want to go for academic research, PhD, postdoc, it is again important for them, because that you are doing valuable research and it will have no value if it is not documented properly. So it is important from that aspect. And then if you have developed something unique, if you have developed something which is good for the human being, so apart from academic research, then it has to go to industry commercialization. So I'm also covering the technology transfer essentials in the student's talk. So I hope everybody is interested now. I'm not hearing anything. Thank you. So quality is meeting established standards. So you have to first establish standards. Then if you meet that, and expectation to satisfy customers' needs. So you have to have the standards first, and then you have to meet that standard. So everything, like for example, if you say, I have a mobile which got bad in very short time, what do you say actually? Quality achhi nahi thi, right? So now we are talking of quality for medicines. So in pharma industry, it is very, very critical to have quality aspects in your development, in your manufacturing, in your quality control. And that is basically nothing but quality or quality assurance. What are the key elements of quality in pharma industry? The product has to be safe because you're going to patient. So the product needs to be safe. The product needs to be efficacious. Otherwise, there's no meaning. If I am giving a paracetamol and it doesn't work, then there will be no impact. So it has to have the efficacy, purity and potency. It should not give me any other side effects, fever, etc. So it has to be having proper purity, proper potency. The product has to be stable. Like for example, now we are moving from pharma to biotech, and the biotech products are not very stable. So you have to maintain cold chain. So if the product is stable, then only you will have a proper effect of the product. The product as well as process has to be consistent. And then we'll also have a GXP, that is good documentation practice, good laboratory practice. We'll learn about that. What are the issues with non-standard product? First is safety concern. So you'll get a news out that X company, cough syrup, there was a problem and India lost a lot of business, etc. The company was like closed for a few days. Then health risk. It is a direct impact on the health of the patient. We are not talking of toys here. We are talking of medicines. We are talking of medicines for healthy people, or even medicines for patient who are sick. So the health risk is imminent. If you have any problem in quality, health risk is a big thing. Customer dissatisfaction. If you didn't like the taste of any medicine, you will be complaining that. So that if you have a proper taste of say Vicks, and this add something and the taste is not good, you don't like. So that is customer dissatisfaction. Then legal and regulatory non-compliances. Suppose there is a guideline and you don't follow that guideline. So there is a regulatory non-compliance. There are recalls and liabilities. So if the product is of substandard, you will have to recall the product from the market. So you manufacture 1,000 units or maybe 1 lakh unit and you put in the market. After a while you understand that the product is not having standard, somebody complains, you again test it and you understand that it is not a good product. So you have to recall. So whatever 1 lakh drugs units that you have sold in the market, you have to get back. You have to give that information to regulators. And then there are liabilities, consequences. If the regulator knows about it, there will be fines, there will be action. You will get like your plant can be closed. Then supply chain disruption. So because of this, like what happens is, if your plant is closed, then you can't supply the product to the market. So there is supply chain disruption. What will happen is competitors will take that share of yours, and it will be very difficult for again you to regain that share. And reputation damage. So imagine you have a company and you are the MD of the company, and there is a newspaper that because of this 10 patients died, or because of this few patients got sick. So you don't want that bad reputation, right? So now understanding quality, let's go to understanding discovery and R&D. So discovery is unlocking novel compounds or therapies to elevate suffering and enhance human health. So everything has to do with respect to pharma is human health. Research is, so discovery is what you can also do in academics. People have labs. Industry also have labs to do a lot of discovery. And then once the molecule is discovered, then when the research and development starts. So research is a scientific investigation aimed at discovering and applying new facts, techniques, and natural laws. And development is a industrial context. In an industrial context, is the work done to finalize the method, process, specs. So once you are in development, you have a proper product and you have a vision that, okay, it will go to non-clinical studies, phase 1, phase 2, phase 3, and then commercialization. So if all passes. So let's look at the cycle, product development cycle. So this is the cycle. It starts with discovery, then research and development. This is called as pre-IND phase. So IND is where you do application, investigation, new drug application. And then the IND review, when you do the application and when the regulators, either in India or US, regulators review, it is called IND review. And then you get phase 1 approval. After that, you submit the data. Then you phase 2, phase 3. Marketing application is called as BLA or NDA in case of pharma drugs, and then post-marketing phase. So now, to speak about Zydus a little bit, we have 13 products approved in Indian market, biologics, all proteins. We have 4 molecules in clinical development, and we have 9 molecules in R&D, and 3 molecules in the discovery part. We are doing a lot of novel biologics. We have one product which is Twin Rab, a rabies monoclonal cocktail, which is approved in India as a novel, and rest are our biosimilars. Now, R&D, in R&D, the engine is, R&D is the engine for the new product development. So it is very important that your engine, that is R&D, has to be very, very strong. So to talk about Zydus a little bit, we have 144 people working in R&D and who are focused on only biotech research. Animal people, clinical people are apart from that. And we have, I'm leading the quality assurance team apart from the regulatory, and we have 7 people who are working only for quality assurance for the R&D biotech research. Now, with respect to GXP, what are the guidelines in product development? So, discovery doesn't have that much guideline, but I would say that good documentation practices is applicable for discovery as well. Because if you properly document, then you can take that product from discovery to R&D. Of course, there has to be scientific merit for that product. Then in research and development, there is good documentation practice. Again, WHO plays a role there. We have 3 guidelines that will come in the next slides. Animal studies, there is a guideline called GLP of USFDA, and all the GLP guidelines of 21 CFR 58 of USFDA and Schedule L1 of India is applicable. For clinical, everybody should be aware of good clinical practices, and there are guidelines like E6 for that, ICH E6. Then when you start with clinical development, you start with good manufacturing practices and good distribution practices, because you have to distribute once the product is manufactured, you have to distribute to various clinical sites. So the distribution starts. So ICH Q7 is talking about clinical trial, and Schedule M is the GMP guideline of India. And then when you are in a manufacturing and QC setup, when you want to commercialize the product, that's where the GMP and GDP starts. And here Schedule M is applicable for the India, and 21 CFR 210 and 211 for USFDA. When the clinical starts, we call it IND. In India, we call it also CTA, that is CT application. When you apply for marketing authorization, it is called BLA or marketing authorization application. So what are the different stages of biotech product development? Everything starts with clone development. So you have to insert the gene of interest into a proper clone, either E. coli or CHO, and then things start from there. You have to do a lot of analytical development to make sure that we have robust methods. Then we have to also do process development for drug substance, process development for drug product, and also once everything development is completed, you come to a end where we say that finalization step. So we have finalized the method, process, and even the specification. So all the development activities are done in R&D. Some of the activities you can do a scale up in the manufacturing, but most of the things are done in R&D. And Stephen Covey's phrase is very important here: begin with the end in mind. Especially when it is biosimilar. Biosimilars are nothing but generic forms of innovative product. So you have to make sure that your product is fixed. The innovative product is fixed, and you have to make a biosimilar of that. So you have to begin with the end in mind, that I have to have that product. If you have a better product, it is of no use. You have to do a lot of studies if it is a better product. If it is a bad product, anyway, nobody will approve your product. So, and quality by design approach is very important. So once you finalize the methods and process and specification, you go for consistency batches. So you have to prove that your product process is consistent and you get a good yield and good product. And then you do product characterization. which are like 30 plus test, primary sequence, secondary structure, primary structure, all those kind of thing, till the last, you can say biological assays, or you can say your SPR assays to prove the biological activity and receptor binding. Then once you prove all, then you have to do non-clinical studies, that is pharmacology and toxicology studies, to prove that yes, your product is working as desired in animals. So once you have some proof of concept there, then is what you go to clinical trial, phase 1, phase 2, phase 3, which Dr. Raghavan has also discussed. Then you file an MA submission and you get marketing authorization, manufacturing launch, manufacturing licenses, and then you launch the product. And you see that this product was launched in newspaper, and there's a launch party thereafter. So all these clinical trial batches and commercial batches has to be manufactured in GMP plant, and the testing has to be done in quality control environment. So for that, you have to basically transfer the technology from R&D to manufacturing, and we'll discuss some part of the tech transfer there. And quality assurance is critical in all these activities. So anybody, can I have raise of hands of people who want to join industry after masters? Wow. So quality, either you're going to research or you're going to manufacturing, QC, everywhere quality is the thing. I would request Professor to share the guideline slides with the students so that they can start reading, because when they come for interview, the first question is going to be what information you know about the quality. So it is, and people who know more on quality, it is good for us to take that person. Now, why criticality? Why quality is critical in R&D? So accuracy and reliability of data. So industry doesn't want to waste money. So if you want to do one experiment, you have to make sure that that experiment is designed properly, gives proper results, so that you can conclude on that one experiment. If you want to repeat, it will be a costly affair. It will be using a lot of resources. We have to also do risk management, whether this experiment is required or not. So you have to do risk management tree and then understand what is required. So it is important for reputation and trust. If somebody says that yes, this R&D is good because they have quality assurance department, they take care of the review of data, they take care of equipment calibration, equipment qualification, their people are trained well. So that is the reputation. You'll get more customers, you'll get more stakeholders, you'll have competitive advantage, and that's a good thing to have long-term sustainability. It is also important that you should have some quality in also academics. Like for example, you may not have QA department, but some of the people who are good in system designing can be given an additional charge of quality control, quality assurance, so that they can set systems. Like for example, when I was doing my masters in Australia, there was one person who used to say, who used to check all our procedures. So I used to prepare a procedure for, say, Spectro, I will do this, and he used to check. And he used to say, harmonize between all the students, so that the data which is generated, the professor or the guides, they can rely on that data. Otherwise, it is difficult to rely on any data. Increase collaboration. This is very important. So quality in R&D can maximize chances of success as well as create value. Now, as I told you, whatever we do in industry, academics is documentation. And what does it? It ensures transparency, reproducibility, and compliance. So like, for example, when you're doing MSc experiment, I know I also have copied the laboratory notebook of my classmates, so I understand what we do actually. But imagine that if you are doing a real experiment and you are not given any data, will you remember the next day? So that is nothing but documentation. You have to do as we do. You do something and just document that thing. So that is very important, and this is very, very critical for R&D. If a person in manufacturing and QC doesn't do proper documentation, that person is put on bench for some time. He can't work. That's the severe punishment that some people are fired because of data integrity. That they don't do proper things, don't report, they report second day, etc. People are fired in industry. In Zydus also we have fired people for that. So let's see the importance of quality documentation in R&D. Comprehensive record keeping. So whatever you have done, you have recorded. Version control is there. Like suppose you have a procedure for pH calibration, and then something new pH meter is there. So you have version control on that, or somebody develops a new procedure, you have a version control. Second version is what I have used. So you have to also in your documentation capture all of this. Procedures, you have to set your procedures. Either it is standard operating procedures to how to operate something, or standard testing procedures, how you are going to test. Data integrity we'll discuss in depth. Traceability. So like for example, in US FDA audit or any key regulatory audit, they give 10 minutes for bringing the document. So they will open any file, they will review that file, and when they say, okay, I want to know this data. So if you say that I'm not fetching that data in 10 minutes, that means that data is not available. So they close that point there, that data not provided. So it is that you have to arrange your department, and that's why QA department is very critical, to arrange your documents in such a way that you can fetch the data quickly. In fact, actually in audits, such as big audits, you have a war room, where this is the audit room and the next room is a war room, where everybody is sitting with the data. Okay, this data. So there are people who are facing the audit. There's a technical person who is the facing audit, quality person facing the audit, and there are runners. So runners are nothing but people who are having just a small yellow stick paper. They will write down SOP number this required. They will go and give to the war room people. The war room people will immediately fetch that SOP. Then one QA person will review that online within 2 minutes, that anything missing, what was discussed, that will be on yellow paper. That runner will give just that yellow paper to the person, and that QA person will see, okay. Then there will be person who is sub-technical person. The technical person is sitting inside. Sub-technical person will understand that this was the context, this is the SOP, what are other documents required. So he will immediately fetch for other documents and keep it ready. And then that SOP goes inside. Once the SOP goes inside, the technical person is clear, the sub-technical person is also clear, this is what we have, 3 following documents, sub-documents to that, and that's how the audit is faced. So it is very important that we should have the traceability part. Regulatory compliance, when you are talking of manufacturing, QC, QA, it is a regulatory requirement that you have to have proper documentation. In R&D also, we'll go through the guidelines which clearly mention that. So if an audit comes to R&D, today, the auditor may say, this is a WHO guideline, are you compliant with that? So we'll go through that. Then intellectual property protection is important, because you have to prove, and you have to have evidence proof, you have to have expert proof that expert has gone through the document and he is signing that, etc. And training and education. So this quality documentation training has to be provided very, very thoroughly. There are 3 guidelines, and I would recommend that this guideline should be having as a discussion point in the MSc and PhD students, because that's where we as India is lacking. Because I've gone through various, I've seen India, I've seen Australia, I've seen Canada, so I feel we are lacking in documentation, a big way. Quality documentation contributes to the generation of high quality, impactful scientific discoveries and innovation. So it is not only important for industry, it is also very, very important for your academics. Now imagine a situation where you have a very good research, and you've used one sugar, sorbitol, mannitol, anything, and that you have not documented. And you have got a fantastic patent, and they said, okay, reproduce, and you are not able to reproduce. Or industry wants to pay $1 million to you, that okay, reproduce. And you are not able to reproduce because you don't know whether what was used. And you lose a lot of time just reproducing that experiment. You'll say, okay, give me 1 month. You'll reproduce those experiments and say, okay, it was mannitol, not sorbitol. So that time has to be properly utilized in the first place to document it properly. Oh, sorry. Okay. In industry, we call it if it is not documented, it is not done. If it is like, for example, if somebody says that I have done this experiment, I will document tomorrow, we said, okay, you're not done it. You're faking it actually. So it is very important to do that. Importance of compliance in industry, you have to know the requirements, rules, standards, governance, regulation, transparency, policies and law. Meet the regulatory requirement, then for maintaining data integrity and to have accurate data, assurance of quality, competitive edge you will get because of that, efficient operations and reduce cost and also improve on overall productivity. Culture of integrity will be developed there, and then empowering QA and RA. Again, QA and RA people has to be empowered, because if the QA person can't authoritatively say that this is not right if you are not doing documentation properly, otherwise it is not going to happen. So you have to give a lot of power to the QA people to really go and find, because like in our case, Archie is sitting here. He is also from Nirma Institute and just joined in July, like last year. So she once, when go to the scientist, senior scientist, principal scientist, she should not succumb to the pressure that what are you talking. She should say, no, sir, this is not right. You have to do this right way. That is empowering. So QA has to be important. And I would recommend again and again that even in academics, there has to be a QA department, okay, if you don't have, but QA as a principle in your academics. So what are the different types of compliance? You have quality control, quality assurance, regulatory compliance, health, environment and safety compliance, GXP, we talked about GLP, GMP, GCP, etc. Good documentation practice compliance, ethical compliance. Ethics is very important also when you're dealing with animal research, when you're doing human research, you have to make sure that you take informed consent, you do proper information about what study it is to the human subjects. Or when you're doing animal research, you're not doing anything like, otherwise the world will talk about COVID. And audit. Audit is one of the key things that we have to start understanding as if you want to go visit industry or join industry. It is important that whatever you do in also research, like for example, in R&D we have self-inspections. So that is nothing but what happens is we form a team of, like there are 12 departments and 13 department is regulatory affairs. So all the 12 departments, we take few people who are good in review, ask them to form a team, we educate them with respect to what is required in audit, and they audit other departments. So that, because the more vision or more perspective you get, the more your departmental quality assurance will build up. And then because scientists can observe other scientists' mistake or what are the procedural errors, etc. So that we do it quite routinely. And what are the benefits of compliance? Enhanced market access. So suppose you have just launched the product in India. If you have proper compliance behind you, you can go to emerging market, you can go to US market, you can go to Europe market. So those things can happen. So your enhanced market access will be there. Patient safety is there. You can go and sleep properly without having tension that whether the patient will fall sick or even die. So that is important. Reputation and trust. Your company's, your reputation is at stake if you don't do properly, and if you have compliance proper taken care of, you will have good reputation and trust. And last one is very important, that if you have all your compliance check done properly, you will have revenue, and that's where you will have growth. And once you have growth on your normal things, then you will step up for innovations, do something new. So that will happen only when you have taken care of all the routine things. And that's check compliance, or compliance is very, very important for that aspect. Otherwise, if you are not doing the compliance properly, you will falter, there might be some problems, and you will get engaged into doing that only rather than doing the innovation. So it is important that you have the compliance properly taken care of. Now let's look into the three guidance that are critical for R&D. Schedule L1 is for GLP. So it was introduced in 2010. So Indian government. This is mainly from the perspective of animal facilities and animal experiments, but also important for lab practices. Now they are talking about general requirements, premises, personal equipment, chemical and reagent. So how to handle chemical reagent? Good house keeping and safety, that's for animals actually. Maintenance, calibration, validation of equipment, then reference material, microbial cultures, quality system, internal quality system audit. So self-inspection is mentioned there. Management review. So what it also says is that apart from your self-inspection and apart from your quality checks or lab rounds, your management should be aware what is happening in R&D and how compliance checks are done. So that is also important. Standard operating procedures, protocol and specification archive raw data. Like for example, in case of GLP, 21 CFR as well as GLP says that even the histopathological slides that you have for animals should be archived for 5 years. So that kind of data archival facility you should also have, and storage and archive automation. Then there is another guideline which was guidance provided by Indian Pharmacopoeia Commission on good documentation practice. It is a very good guidance, talks about principles of ALCOA. We'll again discuss that today. Laboratory records, what should be recorded, data collection and data recording, data reviewing and data reporting. Very important for people who are doing PhD, they should have this data reviewing and data reporting. Rectification of errors, handling of missed entries, that also is captured. And the last guideline, but very important guidance came in 2022, that is WHO good practice for research and development facilities of pharmaceutical products. So if you are developing a pharmaceutical product, either in discovery, research or development, you have to have this guideline. This is a very exhaustive guideline. Quality management and quality risk management, it contains actually sanitation and hygiene, qualification and validation, outsourced activities, what you should control. Like for example, if you are involved in some academic research and you want to outsource sample to say NIBSC UK, what care you have to done is mentioned here. Self-inspection, quality audits, personal training, premises, equipment and instrument, materials, documentation, processing and process design. And some part we'll cover today, but not all. Stability studies, analytical procedures, development, technology transfer is also mentioned in the WHO guideline, life cycle approach, and cleaning procedures and validation. Now let's talk about some of the few, 4 or 5, I'm talking about the key resources of R&D and how we will be discussing that. So first is employees. So under employees, like employees are the key assets that you have who are going to do the work. So we have to make sure that they are well educated, well experienced, well skilled, adequate in number, well trained, ensure routine training, external, internal. This could be called as internal training, but we have to also consider external training. Handover. So this is one of the important things which when I was discussing this presentation with Dr. Sanjeev Kumar, he says, Samir, one of the point which is important is PhD student, when they leave, there has to be handover to the next PhD student or the guide, because that is missed and valuable time is lost if somebody wants to further follow up with that experience. So it has to be, that handover is very, very critical. So employees drive business success. And it is actually very, very apt. I was sitting there and I want everybody to go through the quality statement. The first 4, 5 lines, 5 words are very important. To develop high quality professional. Very, very nicely put, and I really appreciate Nirma for that. With respect to equipments, we have equipments which are in production, we have equipments which are in analytics, all require qualification. So everybody must have heard at least IQ, OQ, PQ, installation qualification, operational qualification, and performance qualification. That is what is required in industry. For example, you have taken a HPLC. Now what happens is, typical Indian mentality, the IQ, OQ protocol all always there, and the HPLC, whether Agilent or Waters will say that, okay, there is 3 lakh rupees, I will do this, give all this document to you. And we say, is it working fine? And we say, yes. And he said, ये document की ज़रूरत नहीं है, 3 लाख बचा लेते हैं. But if we don't have that document, in next time he's not going to support us. He's going to say, repair ka kharcha hai. So it is very important that you should spend that time, because quality means you have to spend on for quality, but quality rewards at the end. So ensure calibration of equipment. So if you have a temperature monitoring system, or if you have pH meter, spectro. So in industry, my lab, like R&D biotech labs, 13 labs, we have 1400 equipments. Out of that, we have around 60% requires qualification, and around 90% require calibration. So that is how R&D works in industry, and it is done. We have a proper scheduler. January में इतने calibrate होने वाले. One person, Manish from my team, goes fetch people that this has to be done. If it is not done, he calls me, he calls the vendor, make sure it happens. Preventive maintenance, it's a very important thing. Like your equipment, if it is not done a preventive maintenance, it may get in bad shape, it may stop working, and your all experiment goes for 2 or 3 months shut down. You can't do the experiment because equipment is nahi hai. So in industry, we can't let it allow to happen. So we have to do preventive maintenance. Preventive maintenance cost, because the vendor person will come here, he will look at your equipment, he will say, ये part खराब हुआ है, इसका replacement करना पड़ेगा. So you have to spend. But the main thing is, you don't have any equipment sitting idle or under maintenance. You have to make sure that it is working, so that your experiments are done, your data is generated, your molecule moves forward. And requalification in case of breakdown, if it is out of calibration, you have to make sure that requalification is done. If it is moved from one lab to other lab, you have to do requalification to make sure that it is working as desired. Qualified and calibrated equipment ensures reliable data. So again, like if you look at from the shift from 2 days that you have discussed so far, it's all academics, research and all that. And now I'm only talking about getting the product right from R&D to manufacturing QC, so that you can commercially launch without any problem. So that shift is important when you are having the hat of industry person, when you want to make that product is approved and launched in the markets. Materials, everybody is aware of raw materials, salts, media, solvents, processors like filter, chromatography resins, excipients, which are basically a drug product has 2 components, API and excipients. So excipients could be sucrose, NaCl, polysorbate, lot of sugars. Yeah.
Doctor, you have 5 more minutes.
Sure.
Okay, thank you.
For DS, plastic and glass bottles, so there are a lot of packaging material also, and it is important that you should have packaging material well under control, otherwise your stability goes down. So control on material ensures desired product quality. You also have processes. So as my colleague Ashini might have spoken about the monoclonal antibody process yesterday. So you have to develop a high quality, high recovery, low cost process, because you have to be competitive also in the market, because other player will come and they will drop prices. So you have to be having competitive. Process should be consistent. Process should be validated when you shift to GMP. Continuous monitoring of the process, and various CMC studies are required, like cell bank characterization, virus clearance validation, filter validation, all those kind of studies. In biologics, process is product. So you have to make sure that the process is developed properly, so that the process related impurities, product related impurities are well separated, and you get a pure product as much as possible. Analytical methods, you have to develop the method with a scientific vigor. In analytical methods, we have drug substance release and drug product release, and around 15 methods are there for each product. And for every product, you have to develop those 10, 15 methods. And we have to have that scientific rigor, methodological approach for developing the methods and finalizing the methods. Then record the development, and then standardize the testing procedure. And in industry, we call it standard testing procedure. Qualify the method for confidence of performance and validate. Good method is important for quality evaluation of product. Stability studies also is important. It is important from the perspective of, it is as a backbone of establishing shelf life of the products, and there are various studies required. Now, role of quality assurance. So we have already seen some of the aspect, but let's go through again. Preparation and maintenance of Apex document like GMP, quality policy. You have a mission, vision here, and quality statement there. So we have to have Apex. This is an Apex document. Quality policy, quality statement, etc. And policies. Then support in preparing templates and batch records. So we have close to 4,000 kind of templates, document that are working, like for each method, you have a ATR, then all those kind of things. Review of process and analytical documents. So RAT, this QA team basically goes through all the documents that are generated as a template. Then that template goes to the user. User fills that template, and it again comes to QA. Material, equipment, ensure qualification, calibration, personal ensure training. Then systems. You have to have systems for document management system, quality management system, and training. You have to ensure smooth tech transfer from R&D to manufacturing. You have to do self-inspections, support in external audits, and quality assurance is key to ensure quality of products. So, and that's why the department is very important. Data integrity, I would like to touch base on these two slides. Maybe technology we can skip. The data integrity refers to accuracy, consistency, and reliability of data throughout the product lifecycle. So key components are documentation. Data is accurately recorded in proper document. Data accuracy and precision, validated analytical methods, calibrated instrument are used. Data traceability, clear link between raw data. So raw data, maybe the pH meter, that pH reading that you have written on tissue paper, right? Right? That's not raw data. Raw data means a document where you have specified that pH will be written in this format. Maybe you can capture 5, 7 pH experiment there. Or a logbook which captures the exact data, not on the tissue paper. Tissue paper is not allowed. And that link from that to your final conclusion or report, that link is important, that is traceability. Then data security, which is very important, that unauthorized access, etc. Then quality management system to ensure research activities comply with the regulatory requirements. Validation and qualification we have discussed. Change control to ensure that if there is a change, that change is documented properly. That's why you should have a 1 page, 2 page, or 3 page form that this is what was the specs earlier, this is what the new specs, and the reason for the change. Otherwise, from one person to other person, in industry also people leave. If you don't have that change control document written, nobody will know how it was changed. So it is important to have those quality systems for that. Data review and approval, training and competencies. So data integrity ultimately contribute to the development of safe and effective pharma products. ALCOA principles, Dr. Amit, if I can extend for 5 more minutes.
You can,
I think this is
3 minutes.
Huh?
2-3 minutes if you can.
Sure, sure. So ALCOA principle is nothing but data integrity. This is all the students should pay proper attention for this. A is ALCOA, then attributable, legible, contemporaneous, original, and accurate. And then there are ALCOA plus. So plus is CCEA. CCA is complete, consistent, enduring, and available. So now let's discuss what it is. So attributable means a person, like for example, I have done an experiment, so I have to sign and date it so that it is attributable, that it is this document belongs to Samir Sangitrao. Legible, it has to be It has to be readable. Otherwise, you know that doctor's prescription, nobody will understand what it is. Contemporaneous. Data must be recorded concurrently. As you do the experiment, you record the data. Data must be preserved in its unaltered state, original. So not the tissue paper, okay? Tissue paper is not original. It is the document. I have seen all of this in my previous life, that's why I'm quoting the example, and I see some smiling faces who acknowledge that. Data must be preserved in its unaltered state. Then accurate data must be actual, free from errors. So there's no errors in that document. The data must be whole and complete set. Data is expected in expected order or self-consistent. So like for example, if you have a report, you have to have all this scope, objective, all those kind of things. It is consistent in that approach. Whatever is desired or given by your agency. Durable and readily available. Again, give an example of audit there, that it is readily available. If you say I have to search for 3-4 days, you're gone. Data is not there. It is not traceable. It is off your mind. Okay. Good documentation practices are practices like format of date. What kind of format you're going to use? About time, whether you're saying 14:30 or 2:30. So that has to be consistent. Ball pen, blue ball pen should be used only. Pencil and gel pen should not be used because pencil anybody can erase, and ink, if there's spillage of water, it is gone. Backdating should not be done. What is backdating? I have done the experiment yesterday and I'm writing the records today. That is backdating. In industry, or if you are in regulated, pharma is a regulated industry, it is a crime. Backdating is a crime. Overwriting should be avoided. Signature is considered complete only when the date is available on that. If empty space is there, strike it out and put not applicable. And then there are, so this, even in academics, I would recommend to define practices for the above points. That how will our research lab or our Nirma university area will work on this. That should be defined. Technology transfer essentials, I would only go to the last slide. Technology transfer essential is begin with the end in mind, planning and monitoring by all. I mean to say person who are transferring the technology and who is receiving the technology. Use similar raw materials, equipment, procedure by donor and receiver. Understand what industry requires. Like we have collaborating with some of the people and they are not aware of what industry wants. So when we don't know the language of each other, it's difficult. Like when we say that, have you proven process consistency, they are saying, oh, what is that? So you have to understand that language of industry. Of course, we have to understand the language of academics, but so that the tech transfer is fruitful. Test drive, trial runs with draft documents is very important so that you can correct, you can have amendments of the document for the final runs, communication and documentation. So key takeaway today is quality is critical factor for R&D. People who are joining industry, please make sure that you go through guideline. If it is not documented, it is not done. In biologics, process is product. Data integrity is important for patient safety and compliance. And the last thing is quality assurance elements are critical for R&D success. Thank you everyone. Bye. Open for questions.
Thank you very much, Dr. Samir, for going through this elaborated processes of from the industry point of view. Now, stage is open for the questions. Professor Arora, if somebody can give him the mic.
Yeah, thank you very much. What I see here is that I got my lab NABL accredited long time back, 20 years or so I have had it. I've looked after all this exactly ditto same principles we follow in the academics as well. It's a diagnostic lab, but it's in a government institution, and then once that lab, which is a diagnostic and disease monitoring lab, that acquired NABL accreditation, we had to go through all these quality. They're same. It's same almost, right?
Yeah.
So that culture slowly was also transmitted to my research lab, where students had to do most of the. Although in the research lab there is so much of turnover, new students come in, it's very difficult to maintain
Yeah.
that. But in a lab we could do it, and I appreciate you have brought out so many good things. I think everyone should follow these, and this really brings in good quality, and the accuracy of results are very important. That's where we all work.
Yeah, yeah. I think to just say one line on that, quality is important for everything we do in life, right? So if you don't do validation because you are in academics, okay, don't do it. If you don't do calibration, okay, for important equipment only you do it. But one thing, if you take from today, that I will not do, what do you say, backdating. That is the most important thing you should do, because that doesn't require any money. You have to have a mindset change for that. So I think there's a quality mindset requirement change throughout India, and I'm sure that that will how Modi's Viksit Bharat will look like.
So I used to tell my lab people, NABL accreditation is just certification. It is the practice, how you work, it will bring in a change in your behavior and everything. So now, slowly that thing comes in practice. So then they don't forget anything. They know that they have to do this.
Yep, correct.
Unless we had a new technician whose duty was to maintain the temperature record of my refrigerators and incubators. And one day he came to me and he says, "Sir, I have 30 papers to sign, which is 30 dates of, I have recorded temperature, but could not get it signed." That kind of things, they happen in academics sometimes. As you say, that backdating, so it was kind of clear case of backdating, and also that's very bad. In industry, it is unpardonable. In academics, we sometimes rely. Okay,
Yeah, yeah.
go ahead. So thank you.
Correct.
Thank you.
Any
Next,
other-
any more questions
I
from
think
the students or Dr. Dalai?
please, sir.
A little bit concerned about, say, in industry, discourage repeating the experiment.
Yeah, so for example, when you run an equipment, when you run an experiment with a design of duplicate, triplicate, that's a design. Okay, but what happens is my experiment failed because of not having proper attention to that. That is what is not recommended in industry. Industry says that, okay, you should design it properly, because everything is resources in industry. Everything is money. Like for example, if you are running a MS experiment, mass spec, it's a very costly thing. So if you don't pay attention on what chemical you are putting or what is the experimental design, you may fail it, and then again you spend money. So that is what I meant for that. It's discouraged to go just like that, just खेलते हैं ऐसा नहीं, मतलब just properly protocol लिखो इसको ऐसा. Okay, guys, so I am sure that you people are quality induced today, inducted, and wish you good luck for your quality journey in your career. Bye.
If thank you very, very much. Thank you.
Thank you, sir, for the wonderful talk. I request Dr. Amit to present the token of appreciation to Dr. Amit. Sorry, to Dr. Samir. I'm so sorry, sir. Thank you, sir. I would also request Dr. Nasreen Munshi to felicitate Dr. Amit. Thank you so much, ma'am.